Good morning, student. Good morning, everyone. We are on the topic vegetative reproduction. There are different kinds of vegetative reproduction. Vegetative reproduction by the stem, roots, leaves. Now, I'm taking the reproduction by roots. See, reproduction by roots. In this, there are some roots having stored food in them. The examples are, in this, roots are having stored food in them and they grow vegetatively like sweet potato. The example is sweet potato. Asparagus develop numerous large swollen fleshy roots. They have like this. They have stored food in them. Fleshy roots. These roots contain a large quantity of food inside and having some roots on them. They have stored food in them. Each such single root is capable of giving rise to a new plant. Each single roots, all can grow into a single plant. Carrot is also example of vegetative reproduction by roots. Carrot is a taproot, is a biennial plant. Taproot, it means it has a main thick root and some branches arise from the main root. This is a biennial plant. Biennial plant. Carrot is a biennial plant. It means the life of carrot plant is 2 years. In these 2 years, first year they stored food. In first year they stored food. In the following year, its stem produces flower and seeds which die by the end of second year. In first year they stored food. They stored food. After second year, they start growing into flowers, fruits, seeds, etc. They develop into flowers, seeds, and etc. All the other parts of the plant. Then, which die by the end of the second year. They all die at the end of the second year. Birds produced at the base of the old stem just above the taproot are mean for the vegetative propagation. Birds produce at the base of the stem. At the base of the stem, birds are produced, and these birds help the plant to regrow for the vegetative propagation. This is the process of reproduction by the group. Here is the activity to grow grass by vegetative reproduction. In this, take some pieces of lawn grass and put them in a pot, cover it with moist soil and leave for it 12 to 10 to 12 days, and sprinkle water every day over the soil so as to keep it moist. After a few days, new grass shoots grow out from the birds at the nodes. Here, what happens when grass is grown in somewhere? Here, the nodes are there. In every nodes, grass develop. Roots, and this is internodes. After every internode is grow into a new plant. So we can see the grass grow rapidly. Conclusion of this, lone grass has long internode between successive nodes. These are the nodes and there is a internode. And these nodes grow into new plant, creeping on the surface of the ground. They develop in this way, creep. They are creeping on the ground. From each node, roots are grown out. These are the nodes. And each nodes develop roots. The auxiliary buds at the nodes produce new shoot. There is an auxiliary bud and this grow into a new plant or grass. This is the vegetative reproduction by roots in grass. What are the advantages of this vegetative reproduction? Farmers prefer to grow certain crops by vegetative method. The advantages in doing so are the following. The advantages are this. Advantages of vegetative reproduction. Reproduction by vegetative takes place in shorter time. It required a small period of time, a very short period of time to grow a new plant. New plants thus produce spread very fast in small area. New plants new plant spread fast in small area. They spread fast in small area, and it is surer method of propagation. There is a surety of growing of new plant. It is a sure method. These are the advantages of vegetative reproduction. And one more advantage of this reproduction is all the good character of mother plant are required retained by the daughter plant. Daughter plant has all good character of its mother plant. Daughter plant carry all good character of mother plant. These are the advantages of reproduction by vegetatively. And some disadvantages is also seen here, and they are disadvantages of vegetative reproduction. As all plants developed by vegetative reproduction are genetically identical. The disadvantages is what? They are genetically identical, same. They are same as the parent plant. They are all likely to be affected simultaneously if a disease is spread in the farm. If there is any disease is present with their mother plant, all the plants has that particular disease. There is a disadvantage of this vegetative reproduction. And the next is dispersal of plants does not take place on its own. Daughter plants, so developed plants developed tend to remain nearby and are restricted to particular area, leading to competition for resources. They grow in a same area. All plants grow in a same area and they do not have proper space, and they are leading for the competition of for resources. They grow in same place and tend to remain nearby and restricted, have a particular space and leading to competition. These are the disadvantages of vegetative reproduction. Now, the next is artificial vegetative reproduction. These days, farmer and horticulturists have developed some certain artificial method of vegetative reproduction, such as cutting, layering, grafting, and tissue culture. The next topic is artificial vegetative reproduction. Artificial, to get some particular to fulfill the particular aim. Here is a there are some method of artificial vegetative propagation, and first one is cutting. In this method, the stem is cut into small pieces with each bearing an auxiliary bud. Stem cut into pieces, cut into small pieces with having bud, and the, af- and cut ends are planted in the moist soil, and this pieces planted in the soil, moist soil, after a few days they strike roots and grow into new plants. After some time, whenever they get favorable condition and we sprinkle water and they get all the required things to grow, they grow into roots, shoots, and in the new plant. This method is normally employed for the propagating plants like sugarcane, lemon, rose, etc. This method is used in sugarcane, China rose, lemon, etc. Then layering is the another artificial method. Layering. Layering is the another artificial method. In this method, a portion of one of the lower branch of the plant with an auxiliary bud at the node is bent down to the ground. What happen here? There is a plant. It has its many branches. Bent is its lower branch. Bent. Move towards the ground with an auxiliary bud. This. See what happened here? Deep branch in the soil. In this method, portion of one of the lowest branch of this plant bent to the soil with an auxiliary bud at the node bent to the ground so that the touches the soil. This touch the soil. This touch the soil, and a ring of bark is removed from the portion. Here is a bark. Layer of bark is removed from the soil, removed from this portion, which is then covered with soil. And after that, removal of this bark, we covered it with soil. Some heavy object such a small piece of stone or brick. Put a small piece of stone, brick or anything, put on them to fix in a soil. Is kept on the branch so that it does not come out from the soil. To keep in the soil and do not come out from the soil. In a few days, when the branch give out roots. After some time, when the branch give out the roots, it is cut off from the main branch. When the roots is developed here, it is cut from the main plant, and this will grow into its individual plant, the main plant, and then continues to grow out as independent plant. Same here, method is used for the propagation of plant like mint. Example of layering method is mint, rose, jasmine. These are the example of method, artificial method layering. In this plant, the lower branch of the plant bent down to the soil, and after this remove the first layer bark of this plant and covered it with the soil. Put some heavy object like stone, brick, etc. For some time, when the roots grows in this plant, remove this stone and cut the path its from its parent plant, and then after that it will grow into a new separate individual plant. This method is known as layering, and the example of layering is mint, rose, and jasmine. Now the third method of artificial vegetative propagation is grafting. Is grafting. In such method like rose, mango, guava, a small shoot or bud of a desired variety. In this grafting, we have a particular aim. A desire of variety of plant is taken, intimately fixed on the stem of another plant of the sum of the same and related species. The plant receiving the bud or the shoot is called stalk. We have to take here two varieties. What is the method? Two variety, two desired variety which we want. Two varieties of plant. We require here two varieties of same plant. This here, what is going on? They take a stem. In this, fix on the stem of another plant. Two variety. One is stem fix on the uh, other stem of second variety. Here is the stem of first variety, and we have to fix the stem of another variety on this stem. This is known as grafting, and in this method we have to take two variety of particular with particular desire. Suppose the flower is of red color and the smell is this flower is good, so take two variety. The plant receiving the bud, here is it, the plant, this is the plant which is receiving the bud. Here we have to put one another part of this and this is fixed in the this is receiving the plant, the another one plant. First variety, and this is receiving the another plant, so it is known as stalk. This is called stalk, and the shoot fix on the stalk. This is the shoot of another plant, and this is known as soil. This is known as soil. The variety of this plant is changed to this variety of this plant, and the stem of this plant is fixed in the first variety's stem, and it is known as stalk For a successful graft, it is important to make it successful. It is very important to that cambium layer of stalk and soil must come into very close contact so that growth may continue. The contact of these two parts may very close to each other. They do not have any space in between them. This is the main process of this method, that the cambium layer of stalk and soil must come together very close so that growth may continue. The grafted plant are then bound together with tape and the together with the tape or joint is covered with. After fixing the stalk and soil, it is bound together. It is bound together. Wrapped it with a tape or covered with and put a wax to prevent dehydration and is protected from the bacterial infection, and put some covered with the wax so that no dehydration, no transpiration will take place here, and protected from the bacterial infection also. And to protect from the bacterial infection, after few days, new cells develop and new plants grow. After some time, the layer of stalk and cambium fuse together. After some time, the layer of cambium this fuse together, stalk and soil, and develop into a new plant. This process is known as grafting. In grafting, we have to take two desired variety plant. First, cut the stem for a middle part of this stem to fix the stem of variety second variety, stem of second variety, and in the plant, the fix in the plant is known as stem is known as stalk, and the plant which have to be fixed in the stalk is known as soil. To this should be this should be in very proper manner, because this contact must be fused. So the fixing of soil and stalk is very important for this. After some time, the layer in between the soil and stalk fuse together and dissolve in themselves. After this, they grow into a new plant. The method is known as grafting. This is done in rose plant, mango and guava. And the fourth method of artificial vegetative propagation is micro propagation. Micro propagation. This is the propagation of plant by tissue culture technique. This is done by the tissue culture. In this method, we require some practical and some laboratory work. We require some laboratory work. In a plant, vegetative propagation is not possible in a plant, then tissue of vegetative buds, shoot apex, or any other suitable part of the plant can be used as an explant. We have to take a part of plant as a explant. Explant is the part of a plant which is used for the propagation, for micropropagation. The explant are treated with sterilization chemical to prevent microbial growth. After choosing the explant, it is sterilized. Sterilized means to make it free from germs by adding some chemical on it, and then cultured in a particular nutrient medium. After sterilization, it has to be put in the some nutrient. Here is a nutrient medium. Explant is here. Then close it, close its mouth. It is cotton plug. Explant, and then shoot apex, explant cells, and some, it is solid nutrient solid nutrient medium. There is some nutrients for the explant to grow here. This is. Then after this, add some regulator. We have to add some regulator. It means to promote its growth. We have to add some regulators, which may be hormones or chemicals, etc. are added. The cell is differentiated into plant part looking like a tiny plant. After some time, after adding some regulators or hormones, it grow well into a plantlets. It grow into a, here is a medium, then grow into a plant. It is plantlets. It is known as plantlets. After 4 to 6 weeks, the plantlets are transferred to the soil. After having some particular parts of this plantlet, take it out and plant it into the soil, transfer it into the soil. This can be used to develop and multiply disease-free stock. This is done because of to get the disease-free variety of a plant. This technique is being used to grow orchids, gladiolus, and cranthosisium. This method is used in orchids, gladiolus, and Christothensis. What we have to do? Choose a plant for the tissue culture, and this plant is known as explant. After making it sterilization, it means make it free from the germs and bacteria by adding some chemicals. After that, put this explant into a conical flask or a apparatus in which it has to be grow with a nutrients medium. That particular conical flask has the nutrients medium to grow, divide cell, from the mass of cell, and this mass of cell is known as cellus. After division of cell, we have to add some regulators or hormones or some chemicals on them to get it well to grow in a proper manner. Cells differentiate into plant part like tiny plants. After some time, these cells grow into develop into some part of the plant, and this plant part of plant is known as plantlets. After 4 to 6 weeks, the plantlets transferred to the soil. Take them out and transfer it into the soil. This can be used to develop the